Thank you, everyone, for coming to this Wednesday's Endocrinology Grand Rounds. We are very happy to have Dr. Matt Cave, one of our own faculty, talk to us about environmental obesogens and NAFLD. I think most of you know him well. Matt initially went to undergraduate at Houston, then came to University of Kentucky, where he graduated with distinction, did his residency at WashU, and then came to Louisville as a GI fellow, and then stayed here. He completed his fellowship in 2007, and since then has been an extraordinarily successful clinician, researcher, educator, and has developed a number of programs within the university and also at the VA. He's currently professor of medicine in the departments of medicine, pharmacology and toxicology, and biochemistry and molecular genetics. He's a distinguished university scholar. As you can see, he's director of several things, including the Integrated Health Science Facility at Core, the Animal Model and Biorepository Core, Hepatobiology and Toxicology Center, and the NAFLD Victor Environmental Health Research Consortium. He has been very active in teaching and clinical practice. He has a long list of medical students, residents, and fellows who have been mentored by him over the years. And many of them have gone on to become faculty at different places. He currently is a PI or a co-PI in about 8 or 10 NIH grants, and also has other funding from industry and other sources. And he's been a highly productive clinician and scientist, and we are very pleased to have him here talk about this very important topic. Thank you.
Thank you. Thanks for the kind introduction. It's really good to see so many friends here, and including some of my active collaborators with Dr. Kling and Dr. Clark, most importantly. So I'm going to talk about obesogens and NAFLD today in the context of obesogens and endocrine disruptors. And so I'll going to start with my favorite quote from environmental health science quote from a Kentucky environmentalist, Wendell Berry, who lives up in Henry County, who said, "Do unto those downstream as you would have those upstream do unto you." And so I think that's sage advice. They call it the Kentuckians' golden rule, environmental health golden rule. And so a lot of what I'm going to talk about, at least in terms of the obesogens, which will be the first part of this talk, can be found in this recently published review article that's got everything you ever want to know about an obesogen in it. So it's the most poorly named article ever written, Obesity 2, to distinguish it from Obesity 1 and Obesity 3, which I think are in the same journal. But this has several of the thought leaders in the field, including Bruce Blumberg, who was the senior author there at the end, who coined the term obesogen. So I'd refer you to that if you want to read just one thing. Several objectives. I'm going to talk about several key terms in the field. What does an obesogen mean? I'll identify some of those and discuss their mechanism. Talk about chemicals that influence fatty liver development and severity. And so the reason that these fit is almost kind of two talks, one on obesogen and one on NAFLD, but the chemicals I'll be talking about in terms of NAFLD are chemicals that are basically liver obesogens. So the things that make the liver fat. And so that's how it all kind of ties in. And then talk about a little bit of clinical guidance on these areas, which is still in its infancy, but actually have some recent developments. And so I think many of you in the room are familiar with environmental obesogens. Some may not be, but the concepts the clinicians have known about for a long time. And so are there chemicals that make people fat? And so we are very familiar with this in clinic in terms of antidepressants, amitriptyline, Remeron, nortriptyline associated with weight gain, and then bupropion associated with weight loss. What about diabetes medicines? So that the thiazolidinediones can be associated with weight gain, and the GLP-1s, which I was getting some help with today, are fellows from Dr. Krishnasamy on how to get those for veterans associated with weight loss. So there's medicines that are associated with either weight gain or weight loss. So why can't there be pollutants associated with weight gain and weight loss? And this particularly makes sense when we talk about something like tributyl-10. So this is a boat. There's mollusks and things sticking, barnacles on the bottom of the boat. It's a horrible picture, but anyway, there's this stuff added to paint called tributyl-10, which kills mollusks and barnacles. And tributyl-10 is a PPAR gamma agonist. And so if thiazolidinediones can activate PPAR gamma to increase obesity, why couldn't this junk at the bottom of the ship that gets into the water, why can't it do the same thing? And so definitions, and so obesogens have been defined as environmental chemicals that can affect susceptibility to obesity and help explain the obesity rates. And so these are related to the endocrine disrupting chemicals, which the Endocrine Society defines as an exogenous chemical or mixture that can interfere with any aspect of hormone action. And what I study is fatty liver, which we initially called toxicin-associated fatty liver or toxicin-associated steatohepatitis, which describe the fatty liver associated with these obesogenic chemicals. And then a relatively more recent term, which is broader, is the metabolism disrupting chemical hypothesis. And so these are chemicals that promote metabolic changes that can result in obesity, type 2 diabetes, or fatty liver. And so really the broadest term is the MDC. So some MDCs are EDCs and some MDCs are obesogens, but not all. And some of these can also cause fatty liver. And so probably most EDC or most MDCs that cause obesity are also endocrine disrupting chemicals and also cause increased liver fat, but that's not always the case. And we saw that recently in a study I've been involved with Tim O'Toole, where he's giving microplastic polystyrene to mice, and so they get increased obesity with altered firmicutes and bacteroidetes in the stool, but they don't get fatty liver. They get altered blood glucose and altered blood lipids, but they don't get hepatic steatosis. So that's kind of an exception. And so how did this field evolve? It's still relatively young. And so in this book called Silent Spring, Rachel Carson kind of set the background for it as the tide of chemicals born of the industrial age has arisen to engulf our environment. A drastic change has come about in the nature of our most serious health problems. So she said that in 1962, and then in 1991, there was a conference at Wingspread Conference Center, which is a Frank Lloyd Wright house built for the Johnson family in Racine, Wisconsin. And that's where the term endocrine disruptor was first coined at this small conference in 1991. And it was about a decade later when obesogens kind of developed and came out of that. So Paula Bale Hamilton said, wrote an article called Chemical Toxins Hypothesis to Explain the Global Obesity Epidemic. My first program officer, Jerry Heindel, who's here, kind of integrated that obesogen or obesity hypothesis with the endocrine disrupting chemical hypothesis, which had been around for about a decade, and that was in 2003. And then Bruce Blumberg here in this paper coined the term obesogen, and so that was only in 2006 when that term was coined. And then about this, a few years later, I was independently studying chemicals that made the liver fat. I didn't know know anything about endocrine disruptors, obesogens, Jerry Heindel, Bruce Blumberg, or any of these people, but they kind of pulled me in, and it took me about 10 years to figure out what I was doing, how that had anything at all to do with what they're doing. But they were studying chemicals that made cause systemic or adipose obesity, and I was just doing the same thing in liver. And more recently, within the last 5 or 6 years, have come out this metabolism disrupting chemical hypothesis. And so that's where we're at over time. And where do these endocrine disruptors and obesogens come from? Well, there's a site here called endocrinedisruption.org where you can look up your chemical of interest. And so there's now over 1,000 known EDCs, and there's over 50 known obesogens. There's at least 50 things that impact liver disease, probably many more. And these can come from several categories, including food production or contaminants. So the PFASs and bisphenols, I'm sure many of you are aware of, phthalates. These are classic endocrine disruptors. I'll talk a little bit more about some of these. Air pollutants, including PCBs, PMs, dioxins, metals, etc., from industrial activity, which we can get from air pollution or food contaminant or water contaminant, and then personal care and home health products, cleaning supplies, etc. One of our partners, Loretta Jofflin's been studying benzoic acid, which is the food preservative that's in diet soft drinks and other soft drinks, and that seems to be incredibly, incredibly bad. So we get exposed to these things at home and then also in medical care. And so this was really concerning. I learned about this probably about 5 or 6 years ago, and there was a reproductive endocrinology researcher that was interested in sperm motility. And so he was in Boston, I think at Brigham. I'm not sure which place he was at, but anyway, he was testing, phthalates. He was into phthalates, and so he was measuring phthalate levels in his patients, or in patients that were coming into reproductive endocrinology clinic, and there was one individual that had the highest phthalates ever seen. They thought it had to be a mistake, and so they brought him back, and it was even higher the next time. So they got to talking to him and said, well, trying to figure out who's your doctor. It was Josh Korsnick, who's a gastroenterologist. So the guy had ulcerative colitis, and he was getting medicine called Azicol, which was a 5-ASA, and so which subsequently, during my GI fellowship, went off, or slightly thereafter, they stopped making it. It went off the market, and it came back sometime later as Azicol HD. And so what Azicol HD is phthalate-free Azicol. So the original Azicol had phthalates, and a lot of the extended release medicines had phthalates in them, including things like proton pump inhibitors, and basically all the GI medicines that were extended release had phthalates. So anyway, in case you're interested, there was a double-blind crossover study on patients getting, I think, Azicol and or not, and looking at various sperm parameters showing an effect. And so there's long been known as some association with fertility and inflammatory bowel disease, but it looks like part of that might be due to phthalates in the pills we were giving our patients, unbeknownst to us. So this is from this obesity review article, the Obesity 2. So this just shows various mechanisms that have been associated with obesogenic chemicals, and it's basically any mechanism that you could think of that's ever been associated with obesity in general. There's some publications showing an obesogenic chemical may impact it. I'll talk today about some of the highlighted ones that include receptor-based mechanisms of action, and a little bit on hormone signaling, and then show a little bit of data that Belinda and Carrie and others have generated on epigenetics, which I think is exciting, but a lot of other mechanisms, including microbiome, circadian rhythms, altered metabolic set point, that I'm not going to talk about. But at one basic level, one way to consider at least the adipose effects of the obesogens, we're just looking at things that can increase adipogenic commitment, adipocyte differentiation, proliferation, or uptake. And this figure was one generated by Bruce Blumberg that's in one of these publications. And you can just see that at every different step, there's a list of chemicals that can do each of these things. And then I've already talked about PPAR gamma-based receptor modes of action. And so here's some example of various receptors that are important in endocrinology, like the glucocorticoid receptor, PPARs, LXR, RXR, ER, etc., and list of different chemicals, that can interact with them. And again, another pharmaceutical example like DES, which can impact downstream changes in adipose tissue and weight. And so obesogens can act like hormones in general, so they can have action at low doses. There can be a non-monotonic dose response. The low dose might be worse than the high dose. There can be receptor-based modes of action at things like the estrogen receptor. There can be diet dependence. There can be sex-specific effects. And there can be windows of vulnerability. And so one of the increasingly well-recognized windows of vulnerability is during development, and so gestational exposures to the chemicals. And so these are, various points in the lifespan, and the same exposure may have different effects depending on when it happens. But developmental programming is increasingly important. And this is well known for smoking. And so maternal smoking's been long associated with increased weight gain and or increased weight in the offspring. Many studies here from this systematic review and meta-analysis. So this is incredibly clear. They call it the developmental origin of health and disease hypothesis. But if cigarette smoking can do it, can some other environmental pollutant do it as well? And the answer is, it looks like yes. And I'll show a little bit of data for that later. But this was a relatively recent publication using a relatively new kind of omics called the exposome. So just like you can do metabolomics, transcriptomics, epitranscriptomics, whatever. There's an omics to measure the pollution exposures called exposomics. And so in this study, they did it in pregnancy. So these were paired maternal and umbilical cord samples from San Francisco. And they measured almost 600 different industrial chemicals, in these umbilical cords, from various classes, pharmaceuticals, pesticides, flame retardants, PFAS, et cetera. I put an arrow on these because I'll show a little bit of data on these throughout the talk. Cosmetics, but the scary one is the unknown. So these industrial chemicals weren't previously reported, so they don't know exactly, what they are. And so this just doesn't show that these chemicals are having any effect, but it's showing that they're there. And this is Michelle Lamerle here. So she's published some interesting data over the last several years on the chemical DDT, which was an old insecticide that's been banned for some time, but still present in probably every organism on Earth. And so perinatal, this first one's a human study. And so this was basically the DDT pollution in the grandmother determined the obesity in the granddaughters. And so so this was a longitudinal multi-generational cohort study. And so DDT levels were measured in blood samples from the grandmothers, and then these levels were associated with increased odds for obesity in the granddaughters at age 26. So it was a sex-specific effect. It didn't seem to determine or affect, and I guess I shouldn't say determine, this was just an association. So it's, causality is not proven, but there was a 3.6 fold increased odds ratio for obesity. And then Michelle basically went on and replicated this in mice, and in terms of giving perinatal DDT exposure. And so that changed metabolic rates, decreased energy expenditure, oxygen consumption, thermogenesis, and exacerbated high-fat diet-induced obesity despite unchanged food intake. And so here's some data from that paper where the EE, or energy expenditure, is decreased in the DDT exposed mice, which are shown in maroon, as opposed to the unexposed mice in the green. And so here's how much DDT, people ingest today based on shopping at a Dallas supermarket several years ago. Let me take this first. Hello? Hello? Call them back. And so I wrote the editorial to this study that was published about 2 years ago looking at PFAS. So the PFAS are the chemicals that were in the movie Dark Waters that were dumped from a plant in Parkersburg, West Virginia, into the environment and ultimately made it down the Ohio River, where some of us undoubtedly drank some of those. And so this was a movie about how the lawyer got involved and won a big legal case. And this lawyer was played by Mark Ruffalo, and he came and was a guest presenter at the Enviro Institute within the last month or so. And but anyway, this study was looking at PFAS exposures in moms, and then also followed up their babies. And they were looking at, here was increased odds for having liver disease, which was probably NAFLD in adolescents, pediatric NAFLD. And so you can, this isn't a dose response, nothing else is. So these are the dose of the exposure in the moms and the odds that the babies get NAFLD later in life. And then here was a metabolomic study that was also done in these several different PFAS species in these orange boxes. You can see that the maternal exposures were associated. These were, the orange boxes are levels in pregnant mom, and the circles are the metabolomics in the babies, or they're now adolescents. And you can see that the exposures were associated with altered metabolism in the babies. And they were, I think 12, 13, 14, 15, something like that when these were done. So it is associated with some metabolic reprogramming that's persistent. And this were all very well controlled for exposures later in life that the subjects also had. So to me, this was very scary, if these last two slides. So it was the obesity epidemic, diabetes epidemic, and fatty liver epidemic that I'm seeing. Is this all related to junk that our moms and our grandparents, were exposed to? And is there anything we could do about it, or are we all doomed? I can't take my grandmother's DDT exposure away from her to improve my life. I guess I'm male, but my sister maybe has to worry. I don't know. So it's scary. And how do these things affect dieting? So when someone loses weight, many of these obesogens are lipophilic compounds, and they distribute to the adipose, and the levels actually go up. Now, this looks like something's dropping over time, but these are all represent an increase. So these are different types of endocrine disruptors, persistent organic pollutants, and you can see here that for every kilogram of weight reduction, this thing here, PCB28, it goes up by 4% in blood. And so they're stored in the adipose, and when the adipose shrinks, they come out into circulation. And are you better off where these things trapped in the adipose? Are you better off when they're in circulation? It might go to brain, heart, liver, kidney, pancreas, some other organ. So this suggests that you may be better off when they're in the adipose. And so there's one clinical trial here suggesting that obesogens may explain why diets fail. And so this is one looking at one of these PFAS chemicals, again, from that Dark Waters movie. And so this was a diet, 2-year diet-induced weight loss trial called the Pounds Loss trial. And so they're looking at the impact of this chemical on weight regain. And so they measured the chemical and the resting metabolic rate longitudinally, just like any other weight loss trial. Most patients lost weight initially, and then they regained the weight, within 2 years. And so they found here that the higher baseline PFAS exposures were associated with a greater weight regain, primarily in women, and that the mechanism for this was probably explained by alterations in metabolic rate. So the baseline exposures may impact metabolic rate to promote weight regain later on. So shifting a little bit to liver disease, the cirrhosis death rate and liver cancer death rates have gone up exponentially, and this was even before the pandemic, and they've gone up even more after the pandemic, largely associated with increased alcohol consumption. So you can imagine, if alcohol sales are up, $1 billion or whatever they were, that the liver disease rates have been up too. And so these are some of the clinical manifestations that get me up at night to see. And we don't know how many chemicals are associated with liver disease, but about a third of high volume chemicals are associated with liver toxicity in this NIOSH pocket guide. And the reason that this is so high is that it's been proposed that the liver is important for chemical detoxification, and so sometimes the chemical may be transformed into a more toxic intermediate, which targets the liver. But for whatever the reason, the liver does seem to be a very common target organ. Here's some EPA data looking at non-cancerous endpoints, and over 25% of all chemicals studied had some kind of non-cancerous liver toxicity, and the next closest organ was kidney at only about 15%, so not quite double, but still high. Don't see pancreas here. No thyroid or pancreas on this graph, but I would think they would be high too if they were studied. And so I got interested in this, looking at rubber town workers. And so this was the vinyl chloride production facility in Louisville that was started in 1942. And then in 1974, hemangiosarcoma was reported in 4 workers. And Carlo Tamburo showed in the right as a hepatologist, or was a hepatologist that was recruited to study this problem with NCI support, and this led to reduction in exposure levels in the chemical plant to the vinyl chloride compound. And there's been roughly 30 liver cancers to date reported from this one plant that were related to the exposure, because you just don't get this type of liver cancer unless you have this type of exposure. And then it was in 2010 where we identified the fatty liver that also occurred in the plant workers. So not quite 30, but 26 workers. Here's one that I saw in clinic. Liver is here on CAT scan, and this thing right here is the liver cancer related to the exposure. And this is a picture of a representative biopsy of a different worker with the same tumor. These were called hemangiosarcoma. So this is a blood vessel-like tumor. And so you can appreciate that this is relatively well differentiated when the cancer is trying to approximate little bitty blood vessels. So it's an endothelial cell tumor. And so now we've got multiple environmental health science grants, which really kind of all can trace back into this legacy, from the 1940s. And here's some biopsies that were taken of these rubber town workers showing non-alcoholic fatty liver disease with these little Swiss cheese looking holes here, a lot of inflammation here, fibrosis. And so this worker had cirrhosis. And so the thing that was unique was really the high prevalence of the disease in the workers. There were a small study, only about 25 that had liver biopsies, but 84% of them had fatty liver, even though their mean BMI was only 25.5. So this is I think what you'd expect from a bariatric surgery cohort, about 80-90% with hepatic steatosis. But the thing that was even worse than bariatric surgery cohort is the prevalence of fibrosis, which was 55%, and steatohepatitis, which is 80%. The steatohepatitis is normally about a quarter or less of the steatosis cases. And so it's a very small study, but I think even to this day, it's the highest prevalence of steatohepatitis and fibrosis in any one cohort. And we published a little on some mechanisms. But a couple of years ago, it was really satisfying when this new disease, which we named here, toxicant-associated steatohepatitis, made it into clinical practice guidelines for occupational health. So, going from discovery to society guidelines in only about 10 years. And so there it is on the table of these other histologic liver lesions, the toxicant-associated fatty liver disease and the angiosarcoma, which was really why we got started at UofL in this type of work. And so I described this TASH, and I thought I was smart and named a new disease, but this was known from a long time ago. So I found this old letter written from this famous pathologist, really the greatest pathologist in the history of liver disease, Hans Popper. So we have, Carlo Tamburo hung on to everything, and so he hung on to this letter I found sometime later, dated 1979. So these are the slides that I found in the biorepository when I was a trainee. And so this letter says that Dr. Popper looked at these slides that Tamburo sent to me. He said Mount Sinai, so you can see here Mount Sinai Medical Center, that the first liver shows so-called fatty liver hepatitis, yada, yada, yada. I cannot exclude alcoholic liver disease or alcoholic liver injury. If this is firmly excluded, I would consider toxic injury, and I hope to receive follow-up information. So NAFLD was not named until 1981, and so he was describing this, before the disease was named, and I guess he never got the follow-up information and never really published much on this. So I thought I was smart, but Hans Popper described this same finding, presumably out of some of these same slides, when I was, I guess I was 5 years old back then, so it had been known a little while ago. But that just shows the importance. If you discover something, you should put a name on it and follow up on it. It goes on to say in the letter that he was off to France for a vacation and hoped to receive some follow-up, so he took a vacation instead. So anyway, we've had fun doing this, and we've extended some of that work on volatile organic compounds like vinyl chloride to many other cohorts. And so at our own Superfund Center, which had important renewal announcement today, it'll probably be in the news somewhere buried behind the mayor's race. It's a bad day to get stuff in the news, but Sanjay was being filmed this morning. But we've found some similar associations between the volatile organics and liver disease and residential cohort in Louisville. Some of us just came from VA, and so this is, and you can't avoid this in the news these days for the lawyers, where you ever at Camp Lejeune, call this number to sue, and some of the conditions that occurred in veterans included fatty liver, or at least been reported, and liver cancer. And I worked with a Deepwater Horizon cohort, investigated fatty liver and some of the cleanup workers in that cohort. So it's been a lot of fun to kind of work with some of these special cohorts and takes the daily drudgery out of life. But fatty liver can be caused by many different things, whether it's, but they're all usually exposures, so it's exposure to fructose, exposure to high-fat diet, exposure to beer, exposure to pollution. They all present are associated with similar histologic changes in the liver. You can't determine the cause. The pathways and mechanisms are probably greatly different, but and related to the specific cause, but histologically they look the same. And of course, co-exposures really, occur, so it's hard, other than the chemical workers, to have someone that's, isolated to just one thing. The reality is people probably not eating a good diet, drinking too much alcohol, at least in pandemic, and then exposed to pollutants when they go across the street or drink contaminated water. So all of these things kind of interacting at once. There's many of these things that have been associated, different types of chemicals that are important. This is one level of importance ranking called ATSDR for the environment and humans. So many of them are ranked near the top. One interesting thing that many people probably don't know here is that the World Trade responders actually get a lot of fatty liver, and so that many of them were involved in a liver cancer screening and surveillance project where they get CAT scans as a part of that, looking for lung nodules, and the CAT scan get the little bit of the liver, and so they're able to look at fat in the liver, just like Tree did, I think, in Alexander Lone's study with Dr. Tamburo for NAFLD using CT to measure liver fat, and so many people had that. And I think there's a couple of these. There's actually a dose response on when they arrived, how close they arrived to the collapse of the building. Gave them increased odds, and nobody knows what it is, but somebody did save the dust from the World Trade Center, and they apparently put it on the mouse nose, and that changed hepatic metabolism by RNA-seq. So, and they think that the dust, the 20-year-old dust, isn't as good as the original dust because there's maybe some volatiles or something that outgassed off the dust particle, and it may have lost some of its potency. But a lot of different cohorts. Here's one on Roundup. That's another favorite of the class action lawsuits recently, but it's been associated with fatty liver as well as particulate matter, and a couple of recent studies now as high as 90,000 subjects where particulate matter was associated with non-alcoholic fatty liver disease, which some people have been suggesting a name change to metabolism-associated fatty liver disease or NAFLD, but it's the same thing, and there's interactions with central obesity, alcohol intake, smoking status, and high-fat diet. So it's really come a long way. So when I started, my first paper on this was 25 subjects or so, and so now they're up to 90,000 subject papers. So the quality of work in the field is increasing. So here's an early approach that we took, looking at something called the exposome using NHANES. And so the exposome was kind of in its infancy then, but they measured about 200 or so different pollutant exposures in NHANES. And we were looking at odds ratios for NAFLD using a surrogate ALT-based biomarker, and that was called unexplained ALT elevation. And so we found dose responses here for polychlorinated biphenyls and some DDT-related pesticides here, and then a couple of the metals. And so these are odds ratios, increasing odds ratios for having this unexplained ALT. This was about 2010, and I hadn't heard of the polychlorinated biphenyls really back then, but I found this expert from MSDS from Monsanto who made them that said the consistent finding in animal studies is that PCBs produce liver injury following prolonged and repeated exposure by any route. Liver injury is first, and it's less than those reported to cause cancer. So, you know, Monsanto is the company that bought you Agent Orange, PCBs, nuclear bombs, a lot of DDT. And so they say something is bad, and they admit to it, that it hasn't a bad effect. You could probably believe it. And so we've been studying that some ever since. So, you know, our approach has been to use these epi studies to find out what chemicals may be bad for the liver and then make animal models of them. But you got to be careful with the epi studies because ALT is not a good biomarker for fatty liver. And so this was a paper written by Sally Condon, our fellow, which just got rejected outright today. So we're going to resubmit it to a better journal, I think, but these are kind of scary data, and it's for, I think the clinicians will appreciate it. So this was NHANES data looking, which added FibroScan in the 2017 year, and so FibroScan is an imaging-based biomarker that can detect fatty liver, and so we categorized the 4,000 NHANES subjects, adult NHANES, into those that had steatosis or didn't have steatosis by FibroScan, and looked at the performance of various ALT cutoffs. And so ALT is actually worse than flip of a coin, or as good as flip of a coin at best, which is why there's a coin toss up here, and that's with the diagnosing fatty liver. So at this extremely low ALT cutoff of 23.5, only 58% of those people that had fatty liver had an abnormal ALT. So if you don't have fatty liver, it's pretty good at ruling it out, but it's no better than a flip of a coin. at ruling in those that do have fatty liver. And if you use a higher ALT cutoff, like most people usually use in clinic, 34 for men, 25 in women, it only picked up a third of the cases. So literally, random chance performs better than ALT for picking up fatty liver, and so that's, I think, impacts. We have to be careful when we're doing these epi studies to find out chemicals that are associated with the fatty liver if we're using ALT like we did at the beginning, because we may be missing a lot of cases. But at any rate, we've been involved in the lab and studying these in an animal model. So here's Benrita Wulong's first study looking at a PCB called 153, which is we called it a diet-dependent obesogen. So control diet fed mice didn't get much abdominal obesity with the chemical, but those that had high-fat diet, co-exposure got more, and you can see here on oil red O staining of the liver lipid droplets in red that it was also worse. And here's a review looking at some of the mechanisms for that, and so one that we've been involved in lately is looking at transcriptional reprogramming, which can be due to receptor-based modes of action, signaling disruption, and then more recently epigenetic changes. So here's a paper that Belinda got out recently looking at just, there's various different types of PCBs, and of course, depending on which mixture of these which you expose an animal to, you get different gene transcription, and some of these PCBs will interact with different types of receptors, as seen here. And because of that, we've looked at a variety of knockout mice for these receptors, aryl hydrocarbon receptors, CAR, PXR, and then we've done an epidermal growth factor treatment because the PCBs activate these receptors, they antagonize this receptor, and we could see that this explains some, but not all, of the story in terms of the liver pathology and the gene transcriptional changes. So this is Josiah Hardesty here, and he found out that the PCBs on a docking simulation that they bind EGF binding pocket in the epidermal growth factor receptor, which you think of in cancer, but he later found that it's incredibly important for liver metabolism, has a much underappreciated role there. And so if the receptor-based modes of actions don't explain all the effects, what about the others? And so there's been some papers in a human population looking at epigenetic mechanisms, including DNA methylation. We've studied liver disease in this cohort. And Heather Clare wrote that paper. I think, Barbara, was you on Heather's committee, and so she has been involved with some of that as well. And more recently, we've been looking at if ALT doesn't work, we're looking at other blood-based biomarkers. And so in here, we've developed a new technique using this Aniston cohort of what we call liquid tissue biopsies, where we measure microRNAs and then let IPA tell us what the liver biopsy would look like based on these circulating microRNAs. And so it gave us kidney, liver, and heart toxicities there. And then more recently, we've been validating that in a second cohort. And here's another paper out of Carey's group looking at some of these microRNAs that we found, and others, some of these we found in mice and could validate changes in them or we found in humans could validate some changes in mice, but it only looked like about 15% or a relative minority of the differentially expressed genes could be attributed to microRNA changes. So, we have some of the picture with the receptor-based modes of action, some with microRNAs, but we still don't have the full picture. And more recently, she's been looking at the epitranscriptome and found some changes that were published. And this one was just recently accepted, looking at the M6A epitranscriptome. And so here you can see differentially methylated genes or RNAs across the chromosome. And I don't want to get into all of the detail, but I think for the endocrinologist in the room, the most exciting, potentially most exciting finding is not the PCB effect, but the identification of, gosh, I don't, you remember what it is, like 7 or 8 apolipoprotein RNAs that are regulated through this methylation in this 6A position, which I don't think was published before. And so, and these are regulated or are changed by PCBs as well, but just the fact that they're there, this is a new mechanism of apolipoprotein regulation, which is important to all of us in clinic that treat obesity. And can we improve that? Is this a new statin, an M6A modulator? Is that the next statin? Who knows? So I'm going to finish up on an exposome study. So this is a R01 that I've got with Naga Chalasani, who interviewed for chair of medicine, and Dean Jones at Emory University. And so this was a little bit different. Instead of taking an environmental cohort and adding liver biomarkers to blood, this was a clinical cohort. So we're taking patients that have fatty liver from a liver clinic, and they're all well characterized. And then we're adding exposure assessment to that cohort, and we're asking the question, are the exposures associated with the severity of the disease? Not do they determine if you develop the disease, but if you have it, which I think is a better way to do it, because I think most people acknowledge that, you know, it's diet and lifestyle factors which are causing a lot of this disease, and, you know, not necessarily the exposures themselves, but the exposures can modify, just like, you know, sedentary lifestyle or other, you know, other things. And so we use this FibroScan biomarker that was used in Sally Condon's paper that gives you an imaging biomarker of steatosis and also fibrosis. And so we're looking at exposures that are associated with differing severities of steatosis and fibrosis. And this was a cross-sectional study. And this, I'm going to show you data from the first 150 or so. We've now done over 1,000, including the Flint clinical trial. So that's the NIH phase 2 study of obeticholic acid. So we have longitudinal data on those patients that were treated with or without an FXR agonist. So we just have to analyze it, which is going to be no big or no small task. But we've got in this first data about 150 subjects. We've got the exposomics data, which was done at Emory. So the subjects came from IU. The exposure assessment was done at Emory by mass spec, and you also get metabolomics included. So Dean roughly calls GCMS the exposome data and the LCMS the metabolome data. And so we've got both of those, and we've analyzed them in different ways. If you're interested in the exposomic methods, they were published in this paper here. There were 140 that passed the quality control for metabolomics analysis, and 134 that passed it for exposomics analysis. BMIs were here, 34. About half of them were diabetic, and they had roughly stage 2 fibrosis by the LSM as the fibrosis biomarker, and CAPS the steatosis biomarker. And first, I'm going to show you what the metabolomics data look like. And so it's like a GWAS, but it's called MWAS, so metabolome-wide association study versus the CAP, which is steatosis. And so the CAP goes from 0 to 400. 400 is the max, and this is the liver fat. So, no liver fat to max liver fat. And you can see all the different dots. They measure, I think it's 60, 70, 80,000 peaks, and these are peaks, not all of which were annotated, and some have significance either up or down at various statistical cutoffs. So these are the 11 features which were past the FDR cutoff, and here's a pathway analysis which was taken using all the features that past the 0.05 cutoff. And so these were done in a continuous and then a categorical fashion, just selecting a CAP cutoff for steatosis or not. But in any case, linoleic acid metabolism was the top pathway on that, which is corn oil, which is not surprising because the alcoholic liver disease animal models all require corn oil diet. And then we did the same thing for the fibrosis biomarker, the LSM. And again, we see linoleate as a important pathway for liver fibrosis in the NAFLD patients. And the next is the exposomics data here. And so we found that 7 pollutants were associated with CAP with a P of 0.05. None of them passed the FDR 0.2 threshold. This BDE66 was the closest. Here's these 7 chemicals associated with CAP, and then 12 were associated with LSM, and those are here, or fibrosis. So steatosis chemicals, so you'll see some things that aren't surprising. PBDEs, which are the flame retardants, and the phthalates, which is the stuff that I give my patients with ulcerative colitis without knowing. And then there was a phenol and octophenol, which was new. And some of these were in both. Octophenol was in both, and then there's a PAH called fluorine, which is here. And this was an integrated analysis, so I think these are really cool. This is called XMWAS, is the name of the software that was developed in-house at Emory to kind of integrate this stuff, where you integrate the exposome data, the metabolome data, and then the clinical data layers all into one figure. And so the clinical data layers are this box here, CAP, this is the box for steatosis, and this is the box for liver fibrosis. And then the circles are the metabolites, and these are in plasma. And then the triangles are the environmental pollutants. And so you can see how the pollutants, the clinical parameters, and the metabolism all associate with each other. And so they associate really into, I think it's 5 or 6 different kind of groups, which are color coded. So there's a light baby blue group, a dark blue group, green group, etc. And and they're not all associated with disease severity. So for example, this polybrominated diphenyl ether here is associated with a pretty potent metabolic disruption. A lot of chemicals associated with that, but it's not associated with the disease severity biomarker. So it's disrupting metabolism independent of disease severity. But these chemicals in the blue or in the yellow do interact or are significantly associated with disease severity. So here's one of the PCBs. We did the study because I wanted to find something else to do other than PCBs because I'm getting tired of PCB. But anyway, it keeps coming back. And then here's the fluorine, which was on another slide. So we've now developed an animal model for this fluorine. Here's a nitrosamine. We think of those for, cancer, at least GI cancers, nitrosamines. But anyway, these fluorine, PCB, and diphenyl nitrosamine were associated with the clinical severity. And here's some pathways that were associated with those. Now, if we take the fluorine and we do the MWAS versus fluorine, this is similar to what we did for disease severity biomarkers. I showed you MWAS versus CAP and MWAS versus LSM. This is MWAS versus fluorine exposure. So you can see that all the subjects were exposed to this PAH fluorine, and then the fluorine was associated with significantly some metabolites in the plasma. And then if you do a pathway analysis of these, you see linoleate here again, suggesting that maybe the fluorine that was associated with the NAFLD severity is doing that by changing something in linoleate pathway, since that was one of the top pathways for NAFLD from the MWAS versus disease severity study. So we get a hint of a possible mechanism. So one thing to be aware of, this was really bad guidance, but this was just published within the last couple months. So guidance on PFAS exposures, testing, and clinical follow-up, and this was the National Academies. So they say clinicians should offer PFAS testing to all patients likely to have a history of elevated exposure. And so basically, everybody in the world's got potentially elevated PFAS exposure if you've been reading this. So So, but they give here, so firefighters, and it's been measured in the Ohio River, so we're all at risk for it. People that live near military bases, et cetera. But it's basically everybody's at risk for PFAS, and so they're recommending that clinicians offer that to all people. Never mind, we don't have a lab, that does test. They've recommended nine PFAS be measured in all people at risk. Quest has a panel that offers 4, and they don't give us really a great billing code on how to do that. So it's not practical, but anyway, you should be aware that in case a patient asks, why aren't you testing my PFAS levels? And then if they have high levels, they suggest you add up the 9, which you can't measure at a commercial lab. But if you add them up and they're greater than 20, you should do various things. If they're between 2 and 20, you should do various things. And these are just things like measure TSH and lipids, which we do anyway. So I don't know who wrote these guidelines, but they're obviously not a clinician, because they, recommending that we measure things that we have no test for and no code for, and then do things that we don't need chemical measurements to actually do to begin with. I don't just order lipids in all patient or the TSH in all patient. But I think it may make a difference in the children is kind of the one group that we would normalize be doing that. So I told Dr. Krishnasamy I'll teach you one thing about SGLT2 inhibitors today. And so how do you get rid of obesogens in your patients? And one way you can do it is to poop them out through GI elimination. So you can give olestra or questran or fermentable fibers that will bind them in the gut. Some of those undergo enterohepatic circulation like a bile acid. You can trap them and poop them out, or you can pee them out. And so there's now a clinical trial actually of using SGLT2 inhibitor to pee out pollutants, just like you pee out the glucose. I guess they go passively probably with the glucose. So anyway, I'll end there. I know it went a little bit long, but thanks for your attention. Well, I'm actually 1 minute. Questions? Oh, and I'm supposed to show the event code.
Well, the octophenol was interesting. That was one. Receptor. Well, if no one has a question, hi Matt, not Steve Winters here.
Okay, repeat
from
the
sunny Florida.
Question.
Nice,
what?
Nice. That was very informative. Can you hear me?
Doesn't know that.
Hi, Steve. Carrie just asked
Anyway,
a question.
I learned a lot from that. So
Okay.
exposure to PFAs is one mechanism to explain why some women who are overweight with insulin resistance get PCOS. I wonder if you're aware of the PFA-PCOS connection and whether you believe it.
Yeah, so the question was I aware of the, I think it was PFAS and PCOS connection. Yeah, and so that's I believe one of the conditions that was covered in that National Academy guidance. And I think it's an important one. This is an example of these chemicals that are endocrine disruptors, metabolism disruptors, and potentially fatty liver chemicals. Steve, I'd forgot that you still come to these, or I would have tried to find some SHBG data for you on endocrine disruptors. I'm sure it probably exists somewhere, but I don't have any. Carrie had mentioned something about the octophenol as being something that was associated with, was it feminization of the rainbow trout?
Subsequent studies in rats.
Yeah, so the phenols are interesting. So there's a group from Mayo that's working with the Emory group, another clinical group that does cholestatic liver diseases, primary sclerosing cholangitis and primary biliary cirrhosis. And they've done about 1,000 patients, but they're doing it the same way as us. They're published their first 100 or so, and then they go with their bigger group. This just came out on their group on PBC and PSC patients, and they found a different phenol was associated with these different types of liver disease. So it's like every liver disease seems to have a phenol, its own phenol these days. And this, instead of octophenol, was nonophenol. So, I guess that's one carbon longer. I don't. Okay.
So in that, there were also some of those things negative. So are there things that actually make you lose weight or remove fat from the liver?
Yeah, so that's a really great observation. Where's Chats?
We're on the right.
Okay. Yeah, so that's a great pickup. So there were some positive associations and some negative associations, and so how do you interpret those? So one interpretation is that they might be false positive hits. So we set a p-value of 0.05 and an FDR of 0.2, which means that 20% of the hits would be false positives, and most we chose because we didn't have any of the pollutants exceed that 0.2 threshold. We showed the ones that were in between, and so it's possible that it's possible that all of the ones I showed either up or down are false associations. We're going to know more. Again, we've done about 1,000, so hope it may be a power issue. And so that's one explanation, is that there are false associations. And then the other explanation would be, as you pointed out, is maybe these things are good. So if it follow, if I postulate that there's junk in the environment that's bad for the liver, well, I should be fair and balanced and say, well, it's entirely possible that there's junk in the, maybe there's an environmental PPAR delta agonist or FXR agonist, that's good, for the liver, and maybe that's what these things are.
As common as diabetes. Get this commonly.
Yeah, so I just got back from the liver meeting, and and those were two, I think, increasingly important concepts that are being discussed. Is one, who to screen and how to screen, and there's some recommendations in the journal Gastroenterology, I believe. But all diabetics for sure should be screened, and I think a FibroScan is a pretty good way to do it if it's available. So there was a lot of data on which biomarker should you be using, and the FibroScan's not perfect, but there's a lot of, but it seems like it's pretty good. You can, it's AROC for detecting at least significant disease is probably around 80% or so, which isn't perfect, but it's pretty decent for a screening test, and you can increase it maybe from 0.7 to 0.78 if you add an MRI and do a bunch of other expensive biomarkers. But for cost considerations, it's probably pretty good. And there's so I think that would be a reasonable place to start. And then the second thing, and this is the case for all of the field really for NAFLD, is there seems to me like they're trying to do it like cardiologists do. So hard cardiovascular events, right? So stroke, MI, composite endpoints, and they've got a name for it now, MALD, I believe, MALD, major adverse liver, I don't know, disease or effects or something. MAL. So something, yeah. So something like MACE, like a MACE equivalent for for liver. And so I guess that'll be important eventually on the medication clinical trial. So right now the FDA is, will be presumably approving drugs based on ability to improve liver fibrosis or NASH histology. But those aren't the same things as hard clinical endpoints. And so
what is the current standard?
So it's.
We did some study before, even all of them had normal weight, but we need to.
Yeah, so that's a good biomarker for liver cell death, right? And I think it's a good biomarker for steatohepatitis. It's not a good biomarker for fibrosis. It's one that we use routinely for research, and we work quite a bit with the company. And actually, we had dinner with them while we were there. It's not FDA approved, and they're a small company and probably having money to do the studies that would be required to do that, but they are interested in potentially making it available as a laboratory-derived test, which we could potentially even do here, just offer it as a, I don't know if you're familiar with that pathway to approval, laboratory-derived test. Which is commonly done. So for like hepatitis D, for example, all of those tests are laboratory derived or laboratory developed tests. So anyway, it may be available here at Louisville in the future, but not now. To clarify what part about it?
Because you had that along with the Southern.
Mm-hmm. Yeah, and the yeah, thanks for bringing that up. And that's these are all coming back, so they're still under development, multiple, different permutations of these for NAFLD in the pipeline. So there's the. Yeah, and inflammation, I believe, too. It was an what's it called? Elafibranor, which was a alpha delta dual agonist that was being developed that didn't pan out. But now there's a company that has, I think, a pan, what they used to call glitazars, I guess. They're coming back for NAFLD clinical trials, although the name's changed a little bit. So the PPAR drugs aren't going away. The FXR drugs are like obeticholic acid are having some issues, at least with Intercept keeps publishing one negative study after another. So I don't know what's going to happen there. The TR beta drugs seem to be exciting, and they're going to have their readout for their pivotal study expected any day now, fourth quarter this year. And if that's positive, they being Madrigal Pharmaceuticals, has the lead compound on that, and they're expected to file. So that's looks to be a something that might actually come. So.
Is there any heart connection?
Yeah, I mean, I think that's the.
These are
It's from mice, but I think this would be potentially the kind of thing that we can develop a whole line of research, future line of research on that, because they were apolipoproteins. Not that I'm a lipid expert, but I'd never heard of. I mean, it was like A through, was it L? There was an ApoL, like Apo A, B, C. I know. But there were letters, high letters that I'd never even heard of as being an APO, but it seemed to be conserved against, across, a large number of apolipoproteins that can, they have these, what do you call them, the DRAC motifs that are, kind of, what I guess, by recognition sites, M6A sites. Yeah, and so presumably, I don't know that we looked across all the lipoproteins, but they probably, presumably, have these motifs.
There is a way to do that.
But anyway, I think that that might be something that might be worth pursuing for as a new line of research that should be pursued in terms of cardiovascular health and probably independent of environmental health because it's much bigger than that. But I think that's a really potentially important finding of Belinda and Carrie's work.
Very much.